Hello, today our topic of discussion is cardiovascular embryology from first aid book. Now, beginning it, we have several topics from first aid book, but we will be doing a bit more in detail to know how we reach up to these particular stages of cardiac looping and the septations of the chambers, atria and ventricle both. Because this is a high yield book, they have not discussed the process of gastrulation and even the formation of a heart tube. So we will discuss the process of gastrulation and also the formation of heart tube. So I will begin from this particular picture. Okay, so let me zoom in. Okay, I will begin with this particular picture. Now, everyone knows these processes of fertilization and then how a zygote is formed. Then it is converted into a morula, a blastula. Then a blastocyst is formed because it has a cavity. And then when the implantation occurs in the myometrium, the divisions, cell divisions actually begin in the form of layers that leads to the formation of specific organs. So first of all, it is a two-layer germ cell, which is called an epiblast and a hypoblast. The cavity above this epiblast is called as the amniotic cavity, and the cavity below the hypoblast is called the yolk sac cavity. Okay, then later on, what happens is that this epiblast layer, I should use a pen. Okay. So this epiblast is converted into these three layers, which we call as the ectoderm, mesoderm, and the endoderm. Okay, so the epiblast is converting into these three layers, ecto, meso, and endo. Okay, now our heart is mostly derived from the mesoderm. I must say not mostly, but actually entirely. The only layer from which the heart is derived, all of its structures, is mesoderm only. So in detail, we will study regarding the mesoderm. Now, telling you in this picture, as you can see here, if you'll ask me to draw, let's say, if I draw this little thing in this way, we have this layer of epiblast, and then we have this layer of meso. Okay, and then we have the layer of endoderm. So ectoderm, meso, and endoderm. And here, a very little part over here is the primitive streak. Okay, this primitive streak gives rise to the primitive node at one end, and the rest of the organs start forming from here. Later, it is divided into neural crest cells, notochord, and elsewise that we will study in the neural chapter. Now, for now, I will take you to another very beautiful picture over here. Okay, let's erase this all. Now, as you can see, this lining, blue color one, this blue color one lining is the ectoderm. This yellow color one is the endoderm. And in between is the mesoderm. Okay, now in the center, this black one is a notochord, and you can see the layer of cells over here, which is called as a primary heart field. Now, I will explain you what is a primary heart field. Primary heart field is basically the cells that are derived from the mesoderm. Okay, we have a primary heart field and we have a secondary heart field. So this structure over here is a horseshoe-shaped structure. This horseshoe-shaped structure is called as the primary heart field. Okay, from this there are atria. Atria are developed, and the left ventricle and most of the right ventricle is developed from this primary heart field. Okay, from this all primary heart field. And there is also a secondary heart field which is formed. The cells of the secondary heart field are present over here, and they moved backward. You can't see the secondary heart field in this picture because they moved backward. Okay, and they formed a aorta, pulmonary trunk, some portions of the right ventricle, and they also maintain the position of the heart structures. So just for a small recap, the secondary heart field what structures do we have in a secondary heart field? The structures we have in a secondary heart field is an aorta, okay, a pulmonary trunk, and some portions of right ventricle. Some portions of the right ventricle. So these are the structures for the primary heart field and for the secondary heart field. Okay. For the primary heart field, I told you atrial left ventricle and most of the right ventricle. Okay. Now, if you understand this picture, let's move quickly to the main part, which is, okay. Now here what happens is when you see that here, this endodermal layer moves downward. It will move downward, okay? And all these pink color cells you see over here, this is the cardiogenic mesoderm, okay? These two above layers are collectively called as somatopleura, and the two layers below one are called as the splanchnoplura. So the two above layers are somatopleura, the two below layers are called the splanchnoplura, and this somatopleura are the layers in which the main structures of the heart tube are enclosed. Okay, now this endodermal layer will move downwards, and what happens is you see that these structures come closer and they get fused, and this area, these two you see, these two circles you see, they are called as the endocardial primordia. These endocardial primordia come closer to each other and they form an endocardial tube. Okay, they form an endocardial tube. And surrounding this pink color lining, this one over here, this is the pericardial cavity. This is the pericardial cavity. So in the heart, we have three layers, okay? The outermost layer of the heart is a pericardium, which is a sac, okay? And there is fluid because there are two types of pericardium. One is a parietal one and one is the visceral one. So in between the visceral and the parietal, we have the fluid. Let's say that this area, this cavity we see is what a sac will be, where a sac will be developed and where the fluid will run, okay? And the closer one will be the visceral, and the outer one will be the parietal. And inside will be an endocardium. This yellow color one is an endocardium, okay? And then this pink color one is a myocardium. So if you understand this, that how these layers will form, we can move further. So after all these steps, we have this one clear structure, circular structure over here, which is the heart tube. And you see that this connection over here, this is open, but later this connection was also closed, okay? And when this connection will close, the pericardial fluid can run within the tube layers of the pericardium. Now, I have another picture. I guess this one. But before moving to this picture, I guess. Okay, you can see this picture. In this picture, this is where the heart tube is present. Okay, initially it was like this, and then later it is here. Now, this is the cranial portion. Okay, this area is called the cranial portion. Okay, ignore the writing. So this is the cranial portion, and this is the caudal portion. So, and this part over here will be the head of the baby. So, what will happen that this area, this cranial end will come forwards. It will come forward so that this little body lying over here can move towards the region of the thorax. So this area comes forward, and then this little body goes inside and inward to reach the thorax area, because the heart lies in thoracic cavity. Okay, it does not lies at the head region. Okay, so but then what happens, and then I just want to show you that this little body over here, this little body, if you can see now, the hollow cavity which is left the hollow cavity which is left here. I think I should zoom it. Okay, you see this is structure. This is structure present over here. So this structure is basically the membrane of the diaphragm, the central tendon, the pleuroperitoneal membrane of the diaphragm in the central tendon region. Okay, that is totally adhered to the heart. Okay, if you know the adult anatomy, you will identify this is that structure. Okay, and later on the area will grow, and the liver will also form in this area and other organs of the digestive tract. So you see here the liver bodies are raised. So this little area is the heart tube, and this is the same. Okay, now I should. Okay, if we go to another picture, this one. Now over here, this is the same process. Okay, and this is also the same process. You can see the dorsal aorta. We will discuss it later. And over here, this is the tube. Okay, this is another picture, and this which I told you, a dorsal mesocardium before. This dorsal mesocardium is a connection right in in this stage, but later on in the adult life, in the postnatal phases, this dorsal mesocardium degenerates and there is no dorsal mesocardium. The heart is only adhered to one membrane, which is actually from the diaphragm, central tendon, to the base of the heart. That's it. No more attachments are there. And this huge big space you see, gray color one, all this space, this is the pericardial cavity. This is the pericardial cavity where the fluid will run. Okay, so now you have all three layers. This is the endocardium, then this pink color one is the myocardium, a thick muscular layer, and then this is the closer one to the myocardium will be the visceral one, and the outer one is the parietal one, and in between this, the fluid will run after the degeneration of the dorsal mesocardium. Okay. Now, the first thing that we will discuss from the first aid will be that these embryonic structures giving rise to which structures. So you will see certain words that you will not understand over here if you're reading this book for the first time and you do not have a good pictorial memory and of how these structures are formed. So let's see, we have this word as truncus arteriosus. Okay, you might have an idea to this that truncus arteriosus gives rise to the ascending aorta and the pulmonary trunk. And regarding the bulbar cordus, it gives rise to the smooth parts, outflow tract of the left and right ventricles. Okay. Outflow tract, there is an inflow tract and there is an outflow tract. Outflow tract means to the aorta and towards the pulmonary trunk, okay? And the inflow tract means the blood coming inside. So I will show you in the picture. We have a picture of bulbar cordus also. Then we have a primitive ventricle and a primitive atrium, okay? Now, the right and the left ventricles, and similarly the right and the left atria, both have two types of parts. One is a trabeculated part and one is a smooth part, okay? So the trabeculated part of the left and the right ventricles, both ventricles, the trabeculated part is arised from the primitive ventricle. And likewise, the trabeculated part of the left and the right atria is arrived from the primitive atrium. But the smooth part of the right atrium, which is also called as the sinus venerum, okay, this smooth part of the right atrium is derived from the right horn of the sinus venosus. Now, you might have heard the word sinus venosus before, but you might not know or remember from your first year MBBS life regarding the detailed structures of the sinus venosus, that what does this left horn or right horn means. So I will show you that in the picture. Okay, so just hold on your horses. Then we have the, excuse me. Okay. Now we have this another structure which is called as the endocardial cushion, which gives rise to the atrial septum and also the membranous interventricular septum and the AV and semilunar walls. So please don't freak out. I will explain you everything. I'm just not reading it. Okay, I'm just reading it because to have you an idea when we will study pictures. Then we have right common cardinal vein and right anterior cardinal vein. Okay, so both of these cardinal veins give rise to the superior vena cava. Similarly, in the case of inferior vena cava, we have three cardinal veins: the posterior one, the subcardinal one, and the supracardinal vein. And also, we have a smooth part of the left atrium left. We studied the smooth part of the right atrium by the right horn of the sinus venosus. But the smooth part of the left atrium is derived from the primitive pulmonary vein. Okay, so the pulmonary veins incorporate and give rise to the smooth part of the left atrium, which we also call as the auricles. So let's move to my first and the most favorite picture, this one. Okay. Now, as you can see, this is the picture of primitive heart, and these are the labelings. You can read them, and I will read for you. Now, these two little bodies lying over here, these are not the ventricles actually, but they are presumed to be the left ventricle and right ventricle in the future. Okay, and this little circle over here is the orifice of initial pulmonary vein. So the pulmonary vein has 4 orifices. So initially it is only 1, but then later on it becomes 2 and then 4, and we will study that too. Now, here you can see the cardinal veins. These are the anterior cardinal veins. We have 2 pairs. One pair of anterior cardinal vein above, and likewise one pair of posterior cardinal vein. So the most lateral one is the left posterior cardinal vein. I should use my pen. Now, this is the left posterior cardinal vein. This is the right posterior cardinal vein. So 2 anterior cardinal veins and 2 posterior cardinal veins. Okay, left and right. Here also we have left and right. In the mid, you can see the umbilical vein. So this, I'm writing a U over here. This is an umbilical vein. Left one and a right umbilical. And the most medial one is the white line veins. Okay, so these are the white line veins. Now, what happens? Okay, one structure is left. This is the truncus arteriosus. Okay, now here, this particular area, at this particular area, this is the sinus venosus, and this is what we will be talking about the whole time now. We read in the first aid that the left horn of sinus venosus and right horn of sinus venosus, the left horn gives rise to the coronary sinus, whereas the right horn gives rise to the smooth part of the right atrium. Okay, so now we will see how it is happening. Now, let's read this. Sinus venosus opens in the center of back of the primitive atrium and has 2 horns. Each horn receives 3 veins, just as I told you. Common cardinal from the body wall, umbilical from the placenta, and white line from the yolk sac. Okay. Then, if you look over here in this picture, yes. If you look over here in this picture, I have explained you this one. Okay, now what happens in the next picture? In the next picture, we are seeing the 4 orifices of the pulmonary veins. We have one orifice earlier, like this. Then it is converted into 2. How these circles are formed? They invaginate inside. When they invaginate inside, they multiply. But that's not what you need to remember in your board exams. Okay, that is what for the understanding of your embryology at the early stages. But here in the board exam, you just need to remember that we have the 4 pulmonary vein orifices that will form the smooth part of the left atrium. Okay, so this will be the smooth part of the left atrium, and here this little body and this little body will form the left auricle and the right auricle, which is the trabeculated part. Okay, and as you can see over here, what was this? The outermost vein, it was the left posterior cardinal vein. This vein will degenerate. This vein is also degenerated over here, and later on, after the connection of the mother and the baby is cut, the umbilical veins and the vital line veins also don't function. So here we assume that now in this picture, the connection of the mother and the baby is cut. Okay, and now the baby is all on its own blood supply. So these cardinal veins degenerated, and the regarding the anterior cardinal veins, the left anterior cardinal vein also degenerates. So the whole cell supply is now from the right cardinal vein. And as you see, this is the right anterior cardinal vein. What happens over here when the left anterior cardinal vein is degenerating, it becomes narrower. When it becomes narrow, the blood moves from left to right. When the blood moves from left to right, the right portion dilates more. Okay, when the right portion dilates more, it will accommodate more blood. And this little area is what we will call later as a coronary sinus. Okay, and this also opens directly into the right atrium. Now, this dilated portion of the right anterior cardinal vein, this dilated portion is what will form a superior vena cava. Now, have you understood how a superior vena cava is formed? A superior vena cava is formed when the right anterior cardinal vein dilates. Okay, and why does it dilate? It dilates because the left side one degenerates. Okay, so among the 4, we have 4 cardinal veins in total. Among the 4 cardinal veins, the left anterior cardinal vein and the left and the right posterior cardinal veins all degenerate. The only one which remains for the adult life to be converted into the superior vena cava is a right anterior cardinal vein. Okay, I think you're clear with the formation of the superior vena cava. So we are done with this. Now, let's talk about the inferior vena cava. If we go back to our book, this point is cleared now that the right common cardinal vein and the right anterior cardinal vein, okay, right anterior cardinal vein, Okay, and why we are saying common? Because earlier, even before these pictures, it was one common which gives rise to two, right and left. So the left one degenerates. So we have this right common and the right anterior one. Cardinal vein is forming superior vena cava. So here in this picture, we are done with this point. Okay. Regarding the inferior vena cava, we have a posterior subcardinal and a supracardinal veins. Now, I have not kept the picture of the subcardinal and the supracardinal veins, but I can draw and explain you how it will look like. Okay, I should extend this a little for the screen, and I can draw here. Now, with the red color, I am drawing the outermost one, which is the posterior cardinal veins. Then from the orange color one, beneath Okay, even the lines are not drawing straight with this pen. Okay, so this yellow color one is the subcardinal, and the most inner one, this. This is I'm drawing the anastomosis over here. Sorry for the picture. Now, this pink color one is the supra. Okay, this is the supra. Supra. I am writing SP in short. Okay, this orange color one is sub. I am writing SB in short. And the red color one is posterior. So this picture is all about the downwards area, not the upwards area. And this messy part in the center is the anastomosis. But this is how they are present. You can see the original picture from the Lang book of embryology. So all these three veins will form the inferior vena cava. Okay, and you can remember one name for sure, which you should remember, is the posterior left cardinal vein. This blue color one is the posterior left cardinal vein. Okay, which will form this inferior vena cava. Excuse me. If you will excuse me for a mistake I did here, was earlier I told you that the left and the right posterior cardinal veins degenerates. And then over here, while explaining the inferior vena cava, I said that this is formed from the posterior cardinal vein, but I'm sorry, the posterior cardinal vein is this one, okay? And this middle one was umbilical, and this one was vitelline. If you remember, this was vitelline vein, okay? So I am extremely sorry. The inferior vena cava is basically formed from the right vitelline vein, and I don't know why they have written posterior subcardinal and supracardinal veins over here, but this is wrong. This is wrong. And I would appreciate if you will do some research about it and will tell me, but up to my knowledge, the posterior cardinal veins, right and left, both degenerates, and what remain is the right vitelline vein, which is converted into the inferior vena cava. Okay, as per the diagram. So we are done with the inferior vena cava, and we are done with the superior vena cava, from where they originates. Also, we understand that how the smooth part of the left atrium is formed, and what else we are left with. Okay. So primitive pulmonary vein, smooth part of the left atrium is done. Inferior vena cava, I told you, correct this. Superior vena cava. Then we have, okay, for endocardial question, I'll take you to another picture. And okay, for the, let's do this horns of the sinus venosus. You have already seen the sinus venosus diagram, but over there, the horns were not present. Obviously, the horns will be present even more primarily to all those structures. Okay, and then a primitive ventricle and a primitive atrium, I'll show you. Also bulbar cordus and truncus arteriosus, which gives rise to ascending aorta and the pulmonary trunk. Okay, so let's move to this last picture first, which I saved. I find this good. A short recap of all of this. So the fate will be the right horn. What the right horn will form? Posterior smooth part of the right atrium. Okay. Okay, also the trabeculated part of the right atrium. Okay, please correct this. This is not smooth. This is trabeculated. Trabeculated part of the right atrium, the right horn, will later form this structure. Then the right common cardinal vein, the lower part of the superior vena cava, or you can also say, instead of the common cardinal vein, the more appropriate to know should be the right anterior. Okay. It is hard to write from the laptop. Right anterior cardinal vein, superior vena cava. Then the right vitelline vein. See, right vitelline vein is forming the opening of the IVC. Then the diminished body and left horn. This diminished body, okay, they're talking about this structure over here. This diminished body, or the left horn, we can say, is forming the coronary sinus, okay? This structure, the coronary sinus. So, and the left common cardinal vein, this one over here, which was earlier like this, but this center portion degenerated. Okay, this center portion degenerated. So this structure over here, which is left, is forming oblique vein on the left atrium. Oblique vein on the left atrium. So I think all this fate is clear to you people. Now let's move to another diagram, which is this one. Now here you can see the horns also. Now this is the right horn. This is the left horn. Okay, these structures, these structures, right and left horn. And this portion over here is the atrium. Okay, the atrium. And this is the ventricle. And this is called the bulbar cordus. Okay, and this area over here is the truncus arteriosus. Truncus arteriosus. Not labeled, but I'm labeling it for you. Oh my God, what's wrong with my pen? So D and an A, and we're done. Truncus arteriosus. So again, truncus arteriosus. Okay, and these two present over here, this is the outflow tract. This is the outflow tract. This will later form the pulmonary trunk and the aorta. Please remember, okay, this will later form the pulmonary trunk and the aorta. These are called the outflow tracts. This portion is called the outflow tract. Okay, this portion is called the outflow tract. Now, the truncus arteriosus, the bulbar cordus, the primitive ventricle, the primitive atrium, and the right and the left horns of the coronary sinus. What the bulbar cordus is giving rise to? It is giving rise to the smooth parts, the outflow tract of the left and the right ventricles. Beneath the bulbar cordus was the ventricle. So in up till this picture, this is inside this ventricle. If I do if I cut it in a sagittal section, okay, and we see inside this thing there are no septums. Okay, this is one sac. This is one sac. There are no divisions of the two ventricles over here, neither there are divisions of the two atrium. This complete thing is one sac. But later on in life, this bulbar cordus is helping this primitive ventricle to give rise to the smooth parts of the left and right ventricles. Okay, and the primitive ventricle you see was the trabeculated part of the left and right ventricles, and the atrium, primitive atrium, will form the trabeculated part of the left and the right atria. Okay, and the horns, the left one, coronary sinus, and the right one, the smooth part of the right atrium. Okay, now one thing is left over here, which is the endocardial cushion. So before moving to the endocardial cushion, I want to show you looping, because another topic from first aid is looping. Okay. Looping. Where is my pen? Here. This bulbous cordus area will move down, and this ventricle and atrial portion will move up, and then this atrial portion will move more up. Okay, like this. So like this, the heart will form. So first of all, initially, we had the two endocardial tubes that I showed you in the very beginning picture, the yellow color two tubes I showed you, the two endocardial tubes, which fused into one tube, which I was calling as a heart tube. Okay, you can go back and see. Okay, so those were the 20 days and the 21 days, okay? And then this whole thing which we studied is formed at the 22 days. So in the MCQ, when we find that the embryology of the heart, or the formation or the development of the heart, starts from which days? So you can say 21 and 22 days, or only 22 days, okay? Not before that. And on 23 day, it started to loop. On a 24 day, it moves like this, and on 35 day. What happens if this fails to loop and go to its original position? Then we see a syndrome, which is called as a cardiovascular syndrome, the connective tissue disorder, which I think is a topic of biochemistry in this book. So that is the defect, and the defect is in the dynein. Okay. So I will show you a video of how the looping happens in order to understand you better. So okay. So let's see this. This is a heart tube which is formed by the two tubes, and the truncus arteriosus, well, the cordus, right ventricle, left ventricle, and the sinus venosus. Now the looping is up. Table comes down and it will go up like this. Okay. Now they are also showing you in a different view. These are the two right atria, and this is the atrioventricular canal. Just remember the atrioventricular canal. Just remember the atrioventricular canal because we will study it. Okay. Now, when you see this structure like this, this is a cut over here. Okay. This is a cut over here. Now I will take you to another diagram to proceed further. I think you might understand what is the process of looping. So this dark brown area present over here is the bulbous cordus, okay? And this is the truncus arteriosus, which will give rise to the pulmonary trunk and the aorta. Okay. And you can correlate this diagram with your first aid book in hand. Now you might also have understood that why we say that the bulbous cordus gives rise to the outflow tract of the right and the left ventricles, because you see it is directly connecting to the right and the left ventricles. And now is the correct time to study the partitions of the heart. Now when you see over here that the cardiac looping, the primary heart tube loops to establish left-right polarity. Left-right polarity is what the position they are taking, the two opposite poles, at the two opposite poles, right and left, begins in week 4 of the development. Because in week 3, what happens was the 3 layers of the gastrulation. Defect in the left-right dynein involved in the left-right asymmetry can lead to dextrocardia. Okay, dextrocardia is a classical feature of the cardiovascular syndrome, which is a problem in the primary, ciliary dyskinesia, that the ciliary, which helps in the movements, okay, is not properly attained, and the patient will have heart on the right side. So remember that a cardiovascular syndrome, the classical feature, dextrocardia, it is a primary ciliary dyskinesia problem, and there is a defect in dynein. And all this whole process is what I've explained to you up till here. But the formation of this complete heart is what we will study now. Okay, let's have a quick let's have this quick thing here. regarding the atria, so separation of the chambers. But before reading this, I would like to read what we have already studied. Like we have studied this heart embryology, and now over here you can see the outflow formation. Neural crust and endocardial cell migrations leads to the truncal and bulbar ridges that spiral and fuse to form aorticopulmonary septum. Aorticopulmonary septum, okay? So please remember this word. I will repeat it again. And truncal and bulbar ridges that spiral. We will see the process of spiral soon in a few minutes. Okay, then after this spiraling and the septum in between the aorta and the pulmonary trunk, it will further lead to the formation of the ascending aorta and the pulmonary trunk. As I told earlier, from the truncus arteriosus. The conotruncal abnormalities, now abnormalities, are associated with the failure of the neural crest cells to migrate. Okay, when the neural crest cells will fail to migrate, what will happen? These three problems: transposition of the great vessels, tautology of the fillet, and the persistent truncus arteriosus. Okay, so these three are, we will discuss it after studying fetal circulation in detail. I will discuss these three conditions, what will happen and what will be the classical features. For now, we are only understanding the structural embryology. Now, valve development. Aortic pulmonary is derived from the endocardial cushions of outflow tract. Please remember the word endocardial cushions because we will soon see its picture. The mitral and the tricuspid valve is derived from the fused endocardial cushions of the AV atrioventricular canal. Okay, valvular anomalies. Now again, the defects. Anomalies can be stenotic, okay, regurgitant, atretic, such as tricuspid atresia, or displaced, example, Epstein anomaly, in which it is displaced from its normal position. Okay, a stenosed valve will not let sufficient amount of blood to flow out or in. And a regurgitant, if regurgitance is occurring, it means that the blood, the valve is not properly closed. It is slightly opened, that after the systole, successful systole, when the blood is pushed into the aorta and the pulmonary trunk from the left and the right ventricles, the little amount of blood is flown back to the atrias, left and right respectively. Okay, so regurgitance is a backflow, and atresia means not properly formed. That is what we call atretic. Okay, and stenotic means it is hard, it is calcified, it is lacking the mobility properly. So now I will take you to this picture. Oh, here. Now, this is a coronal, I think this would be a coronal section. Another atria and ventricle is present over here, which I cut it from the between. Okay, so here you can see this word atrioventricular canal that you saw in your book of first aid. So, and I also show you in that video, atrioventricular canal. So, earlier, I need a pen now. I have to draw some things. It is like this. The cavity inside the atria and the ventricle is like this. Okay, let's say this is above is the atriums, and below the blue ones are the ventricles. So the blood in this picture, in this first A picture, the blood can move in all directions. It can mix up from here to here, from here to here, from down up to down, from down to up. Okay, the blood can totally mix because there are no chambers. There are no chambers. There is no interatrial or interventricular chamber. There is no atrioventricular septums, not chambers, septums. So we are lacking the septums, the atrioventricular septum and the interventricular and the interatrial septums. So we need the first thing which we need to do is to make two longitudinal, two cylindrical, two longitudinal, like this, two longitudinal bodies, like this. Let's say this is left and this is right. So this is left heart and this is right heart. Okay. And this is atrial and this is ventricle. But you see, we don't have a division in between them. Okay. So this is what we need to make. So we will go from this picture to this picture then. Okay. How these two chambers will form. Initially, it was one body, one cylindrical body, as I show you. It was one big cylindrical body. But then we wanted it to convert it into two cylindrical bodies so that the oxygenated and the deoxygenated blood remains in their own chambers. Okay, so for this, we have to fuse these two things. You see, this is the ventral and this is the dorsal endocardial cushions. We need to fuse this ventral and dorsal. In the first picture, the space is wide. The atrioventricular canal is wide. But in the second picture, the atrioventricular canal is narrowed because the ventral and the dorsal endocardial cushions are coming closer to each other, and they are fused. After the fusion of the endocardial cushions, this is what it will look like. And this is what it will look like in this sort of a section. Okay, now this little square structure over here is the fused endocardial cushion. This is the fused endocardial cushion. And this fused endocardial cushion will help you making a membranous interventricular septum, because you know we have two types of interventricular septum, a muscular one and a membranous one. So I will tell you how these septums are formed, but first understand this. This endocardial cushion is forming a membranous interventricular septum, and it is forming the atrioventricular walls on the left and the right side. Okay, the mitral and the tricuspid valves on the left and the right side. And also it will help you forming the septum of the atria, but not the complete septum. Okay, so that was what written in your first aid book over here in the endocardial cushion. That it will make an atrial septum, membranous interventricular septum, and avian semilunar valves both. Okay, I think you should be clear now for endocardial cushion. If you have understood this picture, I will take you to another picture. Please excuse me first. Okay, so now it is time to understand how the atrial septum is formed. Okay, so for today, we are left with in this lecture, we are left with the formation of the atrial septum. Okay, from your book, and also the spiraling of the, or I can say the aorticopulmonary septums. So after this, we will study that. Okay. So, my God. Now, here, this is a septum primum, and this is another image of the septum primum. And like this is this section, and then this is this section. So you can understand it. Now, this is the septum primum. This is the septum primum. Okay, the blood can go from the right atrium to the left atrium. Okay, easily, because there is no septum over here. This is the endocardial cushion, dorsal endocardial cushion. Okay, now what happens first is this septum primum comes down. It comes down till here. Not here, sorry. It comes down till here. Okay, a little portion is left for the blood to flow. Okay, it comes down till here. But it does not fuse to the endocardial cushion completely. Okay, I'm talking about the septum primum. Please focus on this. Okay, it will come down, but it will not fuse to the endocardial cushion. After that, what will happen? There, the endocardial cushion will try to fuse to this, but will fail because this septum primum will form perforations. Okay, it will form certain perforations for the blood to flow out. So, you see, perforations represent the developing foramen secundum in septum primum. Okay, you must have read, we have a septum primum, we have a septum secundum, we have a foramen primum and a foramen secundum. So when the septum primum will come down, it will leave this little area opened, which will be termed as a foramen primum. Okay, and after that, there will be perforations into this septum primum, okay, so that a foramen secundum can develop in a septum primum. So here, at the above area, we will see a foramen secundum. I can explain you it in more better ways. Let's see this down picture. What you see is this primum is fused in the middle. And this little area, which I drawed earlier, that this little area which was opened as foramen primum is still present. Okay, and when certain perforations will line up like this, a little area will be left opened upwards, like here. This will be foramen secundum. So we see MCQs regarding foramen secundum and foramen primum, and you should know that secundum is up and primum is below. Please remember this. Okay. Okay. Now, in the last picture, what we see is the endocardial cushion over here grows a bit more, okay, and it closes this septum primum. The septum primum is closed because the endocardial cushion moves a little more upward, and now there is only one foramen present, which is called foramen secundum. Okay, so if we summarize this a little, first of all, we had foramen primum. Then we had foramen primum below, okay, and foramen secundum above. Why secundum is above? Because it is formed by the perforations in the septum primum, okay. But then, alas, in the end, we have foramen secundum left only, and foramen primum is closed, okay. Why and how foramen primum is closed? It is closed by the upward movement of the endocardial cushion, okay, and it is closed because we don't want so many foramens, okay. So this is up till here. Then we have this another picture. So you see, this is the complete is after all these H stems. We just did these 4. So I will zoom it more. And I would like you to see and understand this thing clearly. What happens next? What happens next is you can see this little structure over here in picture D. This little structure is septum secundum, okay. We have talked about septum primum, foramen primum, foramen secundum up till in these 4 pictures, but we haven't talked about septum secundum. Now I will tell you about septum secundum. What happens here is this little septum growing, it will move downwards, okay. It will try and move downwards. This endocardial cushion, which closes the septum primum, moves a bit more upwards, and ultimately this secundum will, this big hole, you see, this big hole over here is narrowed to this small one. That big hole is narrowed to the small one, okay. And the blood will flow, the oxygenated blood from the right atrium to the left atrium will flow in this manner, in a curved manner, rather than in a straight manner. Here the blood is flowing in a straight manner, but here the blood is flowing in a curved manner, okay. And then in more next stages, the little septum primum which was left above was degenerated completely, and this septum secundum fuses with the elongated growth of the endocardial cushion, okay. I can't say this is a septum primum because septum primum is degenerated. The above one is a septum secundum, which is fusing to this is a septum secundum, okay. And this little one is septum primum, which is degenerated, okay. Septum primum, this is secundum, which is fusing to this endocardial area, okay. And this little thing which was left clearly there, this is not completely fused actually. In the intrauterine life, or we can say when the baby is not born inside the body of the mother, that embryo has this little opening which is called foramen ovale, or which is also called as later fossa ovalis. Okay, after its closure, the foramen ovale is converted into fossa ovalis. But this foramen ovale is present in the intrauterine life, and you can clearly see that if this fails to fuse like this, if it fails to fuse like this later on, how it fuses later on when the baby is born, the connection from the mother and the baby circulatory connection is cut off, the pressure in the left atrium rises up. That pressure pushes this endocardial cushion growth to close this foramen ovale and fuse this little piece of meat to the septum secundum. Okay, so you can say that in the formation of foramen ovale, which septum is involved as an MCQ, septum primum or septum secundum. So in the formation of foramen ovale, septum secundum is involved. Please remember this, please. And after the closure, the blood moves directly from the right atrium to the left, and from the right atrium to the right ventricle, and the left atrium to the left ventricle. And the movement of blood in between the two atriums are cut off because of the closure. And that area, particular area, leaves a mark as a fossa, has remains to remind people that here was previously a foramen ovale in the intrauterine life. So let's move to the book. Now, when we move to our book, I would like you to, oh, shit. Oh, excuse me. Excuse me for that inconvenience and trouble. Okay. So now let's read this, because you have already understood by a pictorial presentation. Septum primum grows toward endocardial cushions, narrowing foramen primum. Okay. Septum primum grows towards the endocardial cushion, narrowing foramen primum. Okay, little foramen primum. Earlier, the foramen primum was this big, and then it moves down. Okay, narrowing the foramen primum. Foramen secundum forms in septum primum. Foramen primum regresses. Okay, next step. Next step was perforations. Okay, next step was perforations. Because of the perforations, this is the endocardial cushion. The perforations fuses, and it gives rise to a foramen secundum. Okay, so foramen secundum forms in septum primum. So this is true, right? Because the perforations are present in the primum. And the foramen primum regresses later on, as I showed you in the picture. The foramen primum regresses, and a foramen and the foramen primum over here, it regresses because the endocardial cushion will move up in the picture. Over here, the endocardial cushion will move up. and it is all fused, and the only thing left open is a foramen secundum. So now in the next, number 3, septum secundum develops on the right side of septum primum. So here is the degenerated septum primum, and here is the formation of septum secundum. Okay, as you can see in this picture also, septum secundum. Okay. As foramen secundum maintains right to left shunt. Okay, this is the foramen secundum, which is maintaining right to left shunt. Okay, septum secundum expands and covers most of foramen secundum. Okay, it expands, it comes down, and it covers most of this foramen secundum, and this foramen secundum is now narrowed. Okay, the residual foramen is the foramen ovale, and the narrow foramen secundum, which is left, is forming a foramen ovale. So foramen ovale is formed by the expansion of septum secundum and the narrowing of foramen secundum. So you see, secundum is there, not primum. Please remember this. Then number 5 is remaining portion of septum primum forms the one-way valve of the foramen ovale. Okay, so here, this remaining portion of the septum primum, the upper one is the degenerated part, and the lower one, green color, is the remaining portion of the septum primum. It forms one-way valve of the foramen ovale, so that the blood can flow from right atria to the left atria in the intrauterine life, and after the intrauterine life, when the pressure rises in the left atrium, it gets closed. Okay, so septum primum closes against septum secundum, is sealing the foramen ovale soon afterward because of increased left atrium pressure and decrease right atrium pressure. Okay, that is what I told you. And then septum secundum and septum primum fuse during infancy or early childhood. Okay, forming the atrial septum. So the atrial septum, the complete atrial septum, is formed during infancy or early childhood in some cases. Okay, now we have a clinical problem called patent foramen ovale, which is caused by the failure of septum primum and septum secundum to fuse after birth. Most are left untreated, can lead to paradoxical emboli. Okay, paradoxical emboli. What is a paradoxical emboli? It is a venous thromboemboli entering the systemic arterial circulation, as can occur in atrial septal defect, ASD. So, this was all about the septation of the chambers, and I think I am also tired. What is the problem? Excuse me. Before moving to the last topic of today, I just want you to show a quick picture of this thing. I told you that we have the pulmonary veins. This is the common pulmonary vein, and these are the pulmonary veins. As you see, they will incorporate inside the left atrium. Okay, this is the left atrium, and they incorporate inside. And in the 5 weeks, they are not incorporated, but in 5.5 weeks, it is incorporated a bit inside. Okay, and you can say that they are divided into 2 pulmonary veins, right and left, at around 6 weeks. And also, side by side, this orifice of the sinus venosus. Okay, remember sinus venosus from the picture, the one which was going like this and the diagonal one and the above one was like degenerated. So if you remember, the orifice of the sinus venosus, there was one orifice in the center. They absorb sinus venosus tissue. It is also incorporating inside, and then it formed the valvuli, and later on it is converted into the superior vena cava opening, the sinus venosus portion. But actually, if we talk about the proper embryological structure from which this SVC was raised was the right anterior cardinal artery. Okay, sorry, cardinal and right anterior cardinal vein. On this inferior vena cava from the right white line vein. Okay, so we were talking about this. Now, at the 8 weeks, you see, there are four openings of pulmonary veins. So this was what I wanted to show you. A quick picture. Okay, let's go back to our book and see that we are only left with this one topic for today. We have completed our first page, and from the second page we are left with this topic, and we will be doing the fetal circulation in the next lecture. If you obviously liked or wanted me to do a video on it. Then, now, how the ventricles are formed, how this all muscular interventricular septum is formed, okay, because there was this little part which grows above and forms the muscular interventricular septum. There is no big deal in this to understand this, but there was a little portion which was opened and which was by the endocardial cushion that formed the membranous part of the interventricular septum. But I told you to remember this aorticopulmonary septum, yes, the aorticopulmonary septum. So I told you to remember this because it will form the ascending aorta and the pulmonary trunk. So all of this thing which is written over here, first of all, we will understand in a picture, a proper picture. So please try to focus here, because this is important, very important. Okay. Now this is a picture, initial picture, after the third picture of the looping. Okay, now you see these are the bulbar ridges. Okay, this is this area. This is this area present over here. Okay, so how it will happen? First of all, it will be like this. Then it will go all the way from here to here. Means it is twisting. It twisted to 90 degrees, and these bulbar ridges are over here now. So these are the same bulbar ridges. First of all, they're present over here, then they're present over here, and then it again twisted from here to here. Okay, again twisted from here to here, and again the bulbar ridges are back over here. So why is this twisting take place? Okay, this makes a total of 180 degrees. 90 degrees twisting in step 1, 90 degrees twisting in step 2. So this is a total of 180 degrees twisting. Okay, now you will understand in this picture clearly. This A is aorta and this P is pulmonary trunk in number 3. Okay, number 3 is first. So at this level, at this particular level, I will try to zoom it more. Okay, I will try to zoom it more. Now you can see this at this particular level. This is number 3. At this level, aorta is posterior. Aorta is posterior and pulmonary trunk is anterior. Aorta is posterior. Okay, you see over here, this is the aorta. And this is the pulmonary trunk. This is the aorta. So aorta is posterior and pulmonary trunk is anterior. But at this level, number 2, it twisted to 90 degrees. The aorta is present towards the right side and the pulmonary trunk is present towards the left side. Okay, aorta is present in the right and pulmonary trunk is present on the left. At this level, number 2, at this level, at this level. But then at this level, when it is forming an arch, okay, it again twisted 90 degrees and the aorta is anterior and pulmonary trunk is posterior. Now you see the aorta is anterior and the pulmonary trunk is posterior. So this is what we call as twisting. It is a total of 180 degrees. Now another picture. You will see it better. Here, pulmonary trunk, aorta. First, aorta is posterior. Okay, aorta is posterior. Pulmonary trunk is anterior. Okay, this is PT. Pulmonary trunk is anterior. Aorta is posterior. Then the aorta is on the left and pulmonary trunk is on the right. Then the aorta is anterior and pulmonary trunk is posterior. So please remember this. Let's move to the book. Okay. Muscular interventricular septum forms. Opening is called interventricular foramen. Aortic pulmonary septum rotates and fuses with muscular ventricular septum to form membranous interventricular septum. I told you about the membranous interventricular septum, okay? It is formed from the endocardial cushion by the process of rotation of aorticopulmonary septum and the fusion it with the muscular wall. Then closing interventricular septum is, well, finally closed. Then number 3, growth of endocardial cushions. Separates atria from ventricles, okay? It will grow laterally also and contributes to both atrial septation and membranous portion of the interventricular septum. So this line is important. Then ventricular septal defect, most common congenital cardiac anomaly. Okay, please remember this also. This is a very important MCQ. Most common congenital cardiac anomaly usually occurs in the membranous septum. Usually occurs in the membranous septum. Okay, so this was all. We started it from here we studied cardiac looping, septation of the chambers, atria, and ventricle in very detailed, more detailed than the first aid. Outflow tract obstruction, now you understand. Aorticopulmonary septum. Okay, truncal and bulbar ridges that spiral and fuse to form aorticopulmonary septum, just as I showed you in this picture. This picture. Okay. Okay. Then valve development and heart embryology. So actually it was very little, seems 2 pages, but now I think you will grasp a good concept. Now we are left with only 1 page, which is fetal circulation. I will try to cover this also. And we are left with some fetal postnatal derivatives also that will also be covered in the video and of the fetal circulation. Okay. After that, we will start anatomy of the cardiovascular system as asked by many subscribers. So thank you all for today. I wish and I hope that you like this lecture, and if you want me to cover anything else, do leave a comment below, and any betterment will be appreciated. Any mistakes if I've done, because I'm also a person, I can make mistakes. So if any mistake I've done in this lecture, you can point me out and correct me. I will be very grateful, because it will help other users, other listeners of this video, and I will pin that comment. Okay, folks, students, we will meet in the next video. Thank you all for watching and subscribe it. Thank you.